All right, good evening, everyone. We're going to go ahead and get started. Our guest speaker has a lot to share with you. I first want to start off by thanking everyone who is joining us tonight for our with our special guest speaker. So thank you so much. I'd like to mention just two things. Please make sure throughout the hour that you are on mute. We don't want any background noise. Thank you. As well as if you have any questions that you would like to ask, please put them in the chat, and towards the end of the presentation, we'll have some time for our guest speaker to be able to answer you. If we're not able to get to all of the questions, we will follow up with an email afterwards. So thank you. Now I'd like to introduce our very special guest speaker tonight. He has an incredible presentation for you. Oh, I guess I didn't introduce myself. I'm Wendy Willock with JDRF Desert West chapter. Apologize for that. Back to our guest speaker. He has an amazing presentation and a wealth of T1D research knowledge.
She's not going to listen to any of our advices. Then
he's from our Pacific Northwest chapter, and I'm going to turn it over and he can tell you more about himself. Mr. Jude Restis. Woo! Thank you, Jude.
Thank you, Wendy. Well, good evening, everyone, and thank you for giving me this opportunity. I'm always very excited to share what's going on. I'm trying to give you the latest update. There's lots of very interesting and exciting things to talk about. This presentation could go on for hours. There's so much, and the biggest challenge I have is to try to figure out what to talk about because there's so much going on. Let me start. I'll just tell you a little bit about myself. So I was lucky enough to be diagnosed way back in 1972. That, if I do the math right, that means this is my golden anniversary of having type 1. I've been involved with the JDRF a long time. I've been a research information volunteer, and that's what I'm doing tonight. I was honored a couple of years ago to be asked to serve on the National Research Committee, a lay member of the research committee for the artificial pancreas program. I'm a ride coach. I'm doing, I think it's about my 25th big ride, a new program, and we'll talk a little bit about it at the end.
Sorry, I'm doing it.
Education volunteer, and I'm currently on the board of directors in Seattle. Okay, with that, let's go. Hold on, because there's lots of information. I always like to start my presentations with this chart. It's the development pipeline. You know, we talk about things like clinical trials, and this chart helps me put it in perspective. So I'm going to spend a minute or so and go through this. Typically, when we think of diabetes research, medical research, we're over on the left side here, discovery research, and that's where you think about people in lab coats looking in microscopes. But it's more than that. And in fact, it has to have each part of this pipeline connected in order to have people out at the end, type 1s that need this either device or medical treatment therapy. Every part of this has to be covered. For the first 20 years or so, the JDRF focused very heavily on that left-hand side, the discovery research, and rightly so. But now we're transitioning into the other aspects of this. So the next one over, translational research. So that's where the engineers get involved. So we move from the laboratory to the real world. So let's say, for instance, in regard to an artificial pancreas, the engineers package it and write the software that make it work. And then we move into regulatory approval. So here's where we get FDA or foreign regulatory agencies to approve the technology that we're looking at. And we throw these terms out: phase 1, phase 2, that middle one should say phase 2, phase 3 clinical trials. What that means, and I'll just real quickly cover that. Phase 1 is it could be a trial of one person or maybe a dozen people across the whole United States. And that the intent of that trial is just to show that it's safe, that we're not going to hurt or kill anyone. Phase 2 is a bigger trial, maybe hundreds, maybe 1,000 people. And now we're trying to see, is the treatment effective? And then phase 3, it's a much wider, and we get a range of older, younger, different all different aspects of our society involved to show that it is safe and it works. Once we get regulatory approval, we move into healthcare coverage. And the JDRF has discovered and figured this out, that if we don't work with insurance companies and our advocacy groups don't work with insurance companies, all of this is for naught. So we have to be able to get the insurance companies to cover the therapy or the medical device that we're working on. And then we have to go out and work with doctors and train people, the users, in order to get the best benefit from all of this hard work that we've gone through this pipeline. So, you know, when we talk about phase 1 trials, I hope you remember what's on this chart. All right, so all of these on this chart are the research priorities, and I'm going to go through all of them except for the lower right-hand corner, the training. We'll have to save that for a different day. So I'm going to start by global universal screening. And why is screening important? Well, we have found that up to about 60% of the people that are diagnosed with type 1 diabetes are in a state of DKA. And we've seen that when a screening program is available, meaning we can find them earlier, only about 3% of the people are in the state of DKA. And why does that matter? Well, DKA, reduction in the DKA at diagnosis can follow that person through the rest of their life. So they'll have better long-term glucose control over the rest of their life. They're lower A1Cs and improved cognitive function. Next, there are several modifying therapies, disease-modifying therapies, drugs that we'll be talking about a little bit later that are geared toward finding those people earlier. Current screening programs missed most of the people at risk, and I'll talk a little bit about that on the next chart. But our goal here at the JDRF is to increase the number of people who are getting screened. And in fact, we want it to become part of normal public health policy. So there's really two types of screening going on right now. There's familial and general screening. So I'm sure all of you in the upper left-hand corner, I hope all of you have heard of TrialNet. TrialNet was able to determine that type 1 diabetes starts long before the diagnosis, and it charted the progression of type 1. And one of the very exciting new therapies that was discovered and talked about last year in all the media was discovered and utilized in trials based on screening. Now we go to the cartoons down at the bottom of this chart, and those are more general. Some like FRIDA, F-R-1-D-A, is a German trial where they widespread screening of many people. Because we have found that over 90% of the people that have been diagnosed with type 1 have no family history of type 1. So if we're only doing family screening, we're missing a lot of the population. And a lot of the previous screening programs are skewed towards youth, pediatric population. And the statistic now is that 50% of people who are diagnosed are adults. So as I've already said, we know that the outcomes are much better with the screening program. So the JDRF has started this new program called T1 Detect. I won't go too much into detail, but it's a general screening program. And if you would like to get a screening kit, you could go to the jdrf.org, go to the website, and order a type 1 Detect kit. And if you don't have enough money, the JDRF will subsidize these kits. With disease modifying therapy. So anything that can alter the course of the disease at any of the stages of type 1, and I'll talk about stages in just a minute, but the goal here is to either prevent it from ever happening, stop it from happening, and then reverse the loss of the beta cells. It can either, these disease modifying therapies can either be focused on the immune system, the beta cells, or both. And when we talk about stages, and we'll talk about these again at least one more time in this presentation, typically people are diagnosed at stage 3. So they're already very much compromised in regard to the beta cell mass that they have, but the disease has started long before that, in some cases maybe 10 years before that. So at stage 1, you may detect, we could, if we did a blood test, we could see that there are 2 or more autoantibodies. And with 2 or more autoantibodies, you have a very high probability of becoming type 1, about 100%. Stage 2, now your blood sugars are starting to get to show signs of being abnormal, and you have 2 or maybe more autoantibodies. Stage 3 is about the point where people are diagnosed, and stage 4 is you've now established type 1 and live the remainder of your life with type 1. And these stages are important because some of the therapies we'll talk about are focused at different levels of the disease. So the first thing is trying to turn off this immune system, which is easier said than done. But our goal here is to turn the brakes on of our immune system. So can we slow down the destruction of the beta cells in the pancreas by our immune system? And one of the things that we're trying to do is change the way the immune system recognizes beta cells. Is there a way that we can stop the recognition of the beta cell by the immune system? And then once those beta cells start getting attacked, they become inflamed, and it's this vicious cycle where now the immune system starts targeting them more because they're inflamed. So can we slow down that inflammation? And on the beta cell side, how can we have? So our goal here is to provide something to keep those beta cells alive and increase their functioning. Can we take existing beta cells and cause them to grow new beta cells? Or can we convert other cells, other pancreas cells into beta-like cells that produce insulin? And I know this is a busy chart and I'm going to go through it quickly, but it's kind of important. So beta cells, the top row. So the function of a beta cell is to produce insulin. When we have type 1, the beta cells become stressed, and then they become more attracted to getting killed by the immune system. So our goal is to produce more beta cells and to protect them. The next row down, T effector cells. So this is now part of our immune system, and these are actually the killer cells. So they go out and they fight viruses and threats to our body. That's what they're supposed to do. In a type 1, they see the beta cell as a threat and they attack. And the thing here is that we have too many of them and they're way too active. So our goal is to either disable locally or to slow them down. Next row, T regulatory cells. So now these cells keep the T effector, the killer cells, in check. They're supposed to be this balance between the two. And for whatever reason, in type 1s now, they're unable to protect the beta cells. There's too few of them or they're just too weak. And our goal is to somehow enhance the T regulatory cells. And then the last row, immune deviator cells, and that's kind of a balancing act between the two. And our goal there is to support the Treg cells and to try to slow down the T effector cells. So let's talk about some of the treatments now that we have shown that work. So the first one is verapamil. So this is a previously approved, FDA-approved drug. It's been used for a long time for blood pressure, but we've now shown that it can slow down the killing off of the beta cell. It keeps the beta cells from becoming inflamed. There's a couple current studies going on with verapamil. And the graph in the upper right-hand corner, it's plotting months on the bottom to C-peptide. Now, it's hard to measure insulin production. So C-peptide is a way, it's a proxy for insulin production. So if you have more C-peptide, you're producing more insulin. So the black line is without verapamil, and you can see over the course of a year, insulin production is greatly decreased. With the use of verapamil, we actually increase, and then we're maybe about where we started from. So using the verapamil, we could slow down that attack on the beta cells. And in the lower right-hand corner, you see some pictures of mice pancreas with and without verapamil. And on the right-hand side, you see the greatly increased mass of beta cells. BANDIT is another trial. It's a JAK inhibitor. They've been used in organ transplant. It's a immunosuppressive. We believe they have current, we believe there's potential for them to slow down and stop the attack on the beta cell. So there's currently a trial that's beginning down in Australia to look at JAK inhibitors. It's almost fully enrolled, and we're hoping that this is going to work out well, and we should see some results next year. JDRF worked really hard to get this approved for human clinical trial. And so here's two things that we're trying to work on. One is, can we have beta cells create more beta cells? Obviously, when the immune system is killing the beta cells, we're losing beta cell mass. We know that we can make human beta cells proliferate in the laboratory, but we've not yet been able to do it in a human. And the red text there is some of the danger that we need to protect against. Obviously, we're making cells grow, and if they go in the wrong part or get out of control, that's a bad situation. So we need to be very careful with that. And on the other side here, converting other cells into beta cells. So the body on its own during type 1 and type 2 diabetes tries to get other pancreatic cells to function like beta cells, to monitor glucose and produce insulin. We know that through some gene therapy, we can deliver some signals to the other cells to form like beta cells. Again, we need to be very careful of this approach, and we need to be able to shut it off if things get out of control. So there's a couple examples here of some research that's being done. So this company, Ionis Pharmaceutical, has a technology that can deliver a therapy directly to the human beta cell. So we have a way to get to the beta cell. And then Dr. Mikey Sander, she's developed an agent that can make beta cells grow. Combining these two, JDRF does best. We create partnerships. So we introduce them to each other, and we helped fund some of the research. And we hope, we've got some, I think it shows great promise, that we can deliver specific drugs to the cells in the pancreas, beta cells in the pancreas, that can make them regrow. Here's another example. Professor Paolo at the University of Miami. He's developed a way where they can deliver DNA in this device, basically, that can fold up in a very specific pattern, and it binds tightly to the target molecule. So he can figure out ahead of time how to devise, how to make this fold up so that it only opens, it attaches and opens with a beta cell. So we could deliver very targeted drugs to the beta cells using this technology. So we've got a couple different approaches in work right now. So we've talked about this, the different stages. You remember what that is. Here are some of the disease-modifying therapies that are currently going on right now, and I'm not going to read through all these, but you can see that at each stage of the disease, there's at least one, in some cases many, in stage 3, that we believe could modify the disease. And I'm not going to read through all of these studies, but at the end, there's a call to action at the end of this presentation. I'm going to twist your arm to volunteer to participate in a clinical trial. And here's an example of clinical trials that are available, and you can see some of them are for newly diagnosed people. And I think that is such an important opportunity for people that you might know that are newly diagnosed. Get them into some of these trials, and we can see if they can keep their beta cells functioning for a lot longer, maybe forever. Okay, we're going to move on now to cell therapies. So what are cell therapies? So cell therapies are therapies which use live cells. So what we're working on, and it is very hard on, is to pull people together across academia and industry to develop beta cells and then figure out ways to put them into our bodies. And I'll go through some of the details on how we do that and how we protect those beta cells when we put them in the body. We know today that if we put beta cells in a human that has type 1, so if we had, let's say, a donor, and we took some of the beta cells and we put them into the portal vein, which gets you into the liver, that they would work. They would monitor glucose and they would produce insulin. It's 100%. Today, we know we can do this. The problem is this pesky immune system that's just going to keep killing those beta cells off. You can see the glucose on for this particular study. A person had islet cells transplanted in, and you can see a very flat line blood glucose. The bad news is they have to be on an immunosuppressive so the immune system doesn't keep killing those beta cells. There's two problems with this approach. One is there's not enough donor cells, and two, there's this problem of the immune system that we have to suppress. So in order for this beta cell replacement program to work, we have to either have a human stem cell derived line of cells, unlimited source, or xenogenic from porcine or pigs. And then next I'm going to talk about how once we get these cells, how can we protect them? So as of today, there are multiple companies working on and have shown successful development of beta cells from stem cells. And I'll go into a little more detail on some of these, but for instance, Vertex, I'm sure you all heard the news just a month or so ago about a clinical trial, a very successful clinical trial. So we've got the cells. So now the challenge is protecting these cells. Where do we put them? How do we find the site in the body? Do we put them, I'll talk a little bit about ViaCyte, but they put them in the lower back. Do we put them into the abdominal cavity? Lots of different details that we need to work out here, and that's what many people are working on that the JDRF is supporting. So there's really 4 delivery and protection tactics: encapsulation, scaffolding, immunomodulation, and gene editing. And I'll go through each of these a little bit more. So I like this chart when I talk about encapsulation. So let's say this diver wants to dive in the ocean. There's 2 things we could do to protect him. One is we could kill all the sharks. But if we just miss one, it's not going to help him. The other approach is to put him in some sort of a encapsulation device where the sharks are there, they're as active as ever, but he's protected from that shark attack. Well, when we talk about encapsulating beta cells, we're really talking about the same thing. So there's 3 ways to encapsulate the beta cell. The one, the picture up in the upper right-hand corner, is a macro encapsulation. So basically, it's a porous device that the cells are in. Oxygen and nutrients can get in and out. The glucose can be monitored through the wall of that device, but it's enough of a barrier that the immune cell can't get in and destroy the beta cell. So either we do it in a macro encapsulation or micro or nano encapsulation, where we have either individual beta cells or clumps of beta cells with a very thin layer. But again, the device has to be able to allow oxygen and nutrients to cross that barrier, but not let the immune system. One of the big challenges of putting one of these in your body is that anytime we put something foreign in, we get this fibrotic response, fibrosis. So the body wants to encapsulate it itself to protect the body from this foreign device. And that's one of the challenges we're facing with this process. So I've already mentioned Vertex. So Vertex Pharmaceuticals, just it wasn't very long ago, released results of a trial they did with one person. They only gave them half the dose that they think. So they have this line of human stem cells. It decreased the requirement for insulin by over 90%. Their A1C and C-peptide levels were great, no adverse impact. So the results are greatly promising. This is very exciting work that we know now that the beta cells that we can derive in a lab work as they're supposed to work. The challenge is this person that got this Vertex line of cells had some other challenges, and he's also taking immunosuppressive drugs. So the first half is done, but the second half, the encapsulation, is not yet dialed in. Lots of the big pharma companies, so Lilly and Signalan, are working on biomaterials that are biocompatible and maybe invisible to the body so that we don't get this fibrotic response. In Biosite, maybe a couple years ago, you remember the JDRF, we were very excited about one of their first clinical trials, the PEC Direct. So that was an encapsulation device. It was implanted in a group of people in their lower back. The good news is that it worked. The cells turned into beta cells. They monitored glucose, and they were, looks like they were releasing insulin. The bad news is that the body then encapsulated it with fiber. The most current news, so as of the beginning of this month, February 2nd, CRISPR Therapeutics and Biosite announced that at least one patient has been implanted with genetic modified cells in one of their devices. So we're hoping, I think this one has great promise. I'll show you a little bit about what the Biosite device looked like, the earlier version. So it's maybe about the size of a Band-Aid, the upper left-hand corner there. And you can see what we want to happen. So 4 weeks, 8 weeks, and 18 weeks into this, you can see the body is starting to wrap itself around there, and the blood is exactly what we want to happen. We want that barrier to be able to transmit blood and nutrients. And this is one I'm kind of excited about. My day job, I'm an aerospace engineer, and we've been playing around with 3D printing for quite a while. We can very precisely design scaffolding or things that would hold beta cells. We could put drugs in next to the beta cells. So this, I think, holds some great promise to develop very complex things on a very small scale. So the next way we're going to protect the immune system, or the beta cell, is by immunomodulation. So at a very local level, we can either put a drug in there or tweak something to make the beta cell not visible or less likely to be attacked by the immune system. The third way is gene editing. So can we get in and modify the genes that control the cell function, that the proteins that are produced are not visible or less likely to be destroyed by the immune system? And I've already talked about that trial, CRISPR and the Biosite device that has genetically modified genes. So you might have heard of the JDRF Centers of Excellence. So the JDRF and some prestigious universities around the US have teamed up to focus on specific areas. And here's 3 examples of JDRF Centers of Excellence, and they're focused on gene editing. So we've got some very smart people working on how to make this work. How can we edit the genes? How can we get that beta cell to either be stronger and not be subject to destruction by the immune system, or it'll be invisible to the immune system? So I'm kind of excited about these smart people working on this project for us. And the last way we're going to protect those beta cells that we put in is in some sort of a scaffolding. So think of a sponge that can hold beta cell. It's an open structure, so blood and nutrients can flow in and out, but we could also then put targeted drugs within the other openings in these scaffolds that it would locally deliver drugs to keep the immune system at bay. And here's an example of a company that's working on scaffolds, the Sernova company. They had a clinical trial last year and it showed great promise. And the way this particular device works, they put it in first and they let the body get used to it, the blood vessels attach to it, and then after it's been in for a while, then you would insert the beta cells into that device. So what is the JDRF's role in this beta cell replacement? So one of the things that we do best is pulling people together with expertise in many different aspects of this, instead of having one company work on all of it, coming up with the beta cells and coming up with a device that encapsulates it and figuring out how to get drugs included in that. We pulled together all these people across academia or industry, and we started this consortium where they can all work together. There's companies that are still competing with each other, but they've agreed to the way that this device, certain things that have to be done a certain way. And by doing that, we can go across industry and catalyze additional monetary investment. And then hopefully you've heard of the T1D fund, but it's basically like a venture capital fund where we can provide financial support for these companies. And then we're already talking to the regulatory people. So we're already having discussions with the FDA on beta cell replacement. We're not going to wait till it's done and then go hand them a package for approval. So they know what we're doing and they know which way we're headed. Next chart, not advancing. There we go. And I'm not going to go through all the details here, but here's an example of some of the companies that we've pulled together, introduced and pulled together to solve this encapsulated beta cell project. So there's lots of activity and lots of money being spent across industry. And here are some of the current trials that are available. And again, at the end, I'm going to have a little call to action for you folks to participate in clinical trials. But if you're interested in encapsulated beta cell, there are current trials going on. And then the last thing I'm going to talk about is improved outcomes. So we know today we have good insulin, human insulin, basal, rapid-acting insulin. We have glucagon. Unfortunately, it's typically only used for emergency use. Insulin, SGLT inhibitors, and we know that we have got several versions of hybrid closed-loop artificial pancreas systems. We've got factory-calibrated CGMs. So, when I look back 50 years ago when I was diagnosed, none of this stuff was available. So I'm thinking life is pretty good right now, but we believe there's still a lot to be done, and there's a great promise for things like next generation, much faster, better insulins, other adjunctive therapies, fully closed loop systems. So you could have an artificial pancreas that doesn't require the person to bolus for a meal. CGMs, continuous ketone monitors. Basically, we think there's still things out there that can improve our health and happiness. So glucose responsive insulin, or some people call it smart insulin. So back in the early 2000s, JDRF had pretty much a contest. We threw this challenge out and said, we'd like people to work on an insulin that would respond to glucose. So it's not active when your blood sugar is where it's supposed to be. When your blood glucose rises, the insulin starts becoming active and kicks in and works. We funded a couple different projects, and then some of the big pharmaceutical companies thought they showed great promise, and they partnered with the JDRF and actually bought, in some cases, bought some of the small startup companies that were working on glucose responsive insulin. So right now, Sanofi, Eli Lilly, and Novo Nordisk, we know all have internal glucose responsive insulin programs. And we know that Novo Nordisk has one that's either in or very close to human clinical trial. Artificial pancreases. So I'm not going to read all of the different versions here, but we've got the Medtronic that's been around since 2016. The Tandem first came out with the Basal IQ, and now the Tandem Control IQ. So those have already been approved by the FDA. I'll talk a little bit more about Loop, but we believe in 2022 there's a big influx of new artificial pancreas systems that are going to be available to us type 1s. Just recently, the FDA approved the Omnipod 5. So this is the first tubeless artificial pancreas system. The time and range, we all know the closed loop system or the hybrid closed loop systems work pretty well. So now if somebody wanted to use a tubeless system, this is available to them. We think that in 2022, here's some of the things that are going to happen. Now, we've got the upper right-hand corner, Insulet, check that box. That one's already been approved. And in the middle on the lower row, EverSense, that one, check the box, has been approved by the FDA. So that's a 180-day implantable sensor. Tandem has got their Mobi device, which is a smaller, very short tube device. And instead of having to play around with your pump, it'll use a smartphone. You can control it via smartphone. The electronic device, the 780G, and also now they're working on approval for a 7-day wear infusion set. So instead of every 3 days, you can wear that thing for 7 days. In Tidepool, I'm going to talk about that next. So the JDRF with Helmsley Charitable Trust pulled together the Tidepool, which for those of you might have heard of it as Loop. So Tidepool is now working with the FDA to get approval for the Loop do-it-yourself system as another device that we can use. The real interesting part of this, though, is that this system will be interchangeable. So a user could say, I like the brains of the Loop system. I like the Dexcom CGM, or I like the EverSense CGM, and I like a Medtronic pump. The goal here is to have complete flexibility, that all of these can talk to each other and the user can fine-tune the system they want. So the Loop algorithm, the heart of the Loop system, is currently under review by the FDA, and we're hoping this year that could be approved. And I threw this one in there just because I thought it was kind of neat. Someone discovered that there's this snail that uses insulin as a weapon. It squirts a little bit of insulin out, and it's such fast-acting insulin that it kills or incapacitates its prey. And we're trying to figure out how do they make that insulin and what makes it so fast, because we want faster-acting insulin. So not a study, but I just thought it was kind of cool. This is something that I hadn't yet heard of. So I talked a little bit about some of the technology that's available and some of the things that are in work, but how can you help? How can you help get research from that left end to the user out at the right end? And we've discussed a lot about clinical trials. They're essential, so nothing gets approved unless it's gone through the human clinical trial process. So I have a call to action for you. We know now the JDRF has determined that about 80% of clinical trials are delayed due to either insufficient number of people signing up or not signing up at all, And what that does is it lengthens the trial. So instead of getting a trial done in 6 months, it might take a year. It delays getting answers to us that are waiting for these clinical trials to be completed. And one of the most dangerous things here is that it's a disincentive to the sponsors for these trials. So if a big pharmaceutical company doesn't see progress in a clinical trial, they may pull the plug. So we all collectively, everyone on this call being involved with the JDRF, works really hard to raise money for the JDRF. We're out there doing walks and riding our bicycles and fundraising because we know it's so important to fund these clinical trials. And it's just a shame that 80% of these trials are delayed. So if we want to make this cure happen quicker and these devices and treatments and therapies come quicker to us, we need faster enrollment. So what can you do? So one way to do it is to go to the JDRF.org. So there's a clinical trial match tool at JDRF.org. And within about a minute, you can go through the process. It asks where you live, how far would you like to travel to participate in a clinical trial? Do you have type 1 or do you have a family member that has type 1? You just click through these different options and then it will tell you within your area, or if you're willing to travel. I know in the Seattle area here, we have some world-class research facilities that have trials open right now. So anyways, you click through this thing and it'll tell you if you're able to participate, and then it's up to you to take the next step. And I would urge you to do so. And I'm not going to read through this, but I hope Wendy can make this chart or this whole slide deck available, because there are a lot of resources available. There's another one called clinicaltrials.gov, and you can do a search of any kind of clinical trial, not just type 1, but you could focus on type 1 research. So there's a lot of clinical trials that are available. I showed you a few for some of the encapsulated beta cells. So I strongly urge you to participate in a clinical trial. And with that, I thank you for your attention. And again, I thank you for giving me this opportunity. A lot of exciting research out there that I think is very promising. And with that, if we have any questions, we can try to address some of them.
Thank you so much, Jude. What a wealth of information. Really appreciate all that you've shared with us tonight. Most of, or if not all of the questions that people put on their registration, because I had put a spot there for people to ask ahead of time, you pretty much addressed. So I thank you so much. There's just a couple that from the registration that I noticed maybe have a little more information. And if there's any current or new research on the data of the correlation between becoming type 1 after getting COVID. And there is I will put in the chat a link, but if you want to speak to that.
Okay, yep, so actually Wendy sent me that link, and best I can tell, and I probably should just start by saying, I don't really have a good answer, but best I can tell, reading what the JDRF published, which was research done by someone else, it seemed like in youth, there maybe is a correlation between recovery from COVID, and what it doesn't make clear is it type 1 or is it type 2? So we always have to be careful of that, because when you read the news media, they'll just say diabetes. And I couldn't discern the difference between the two. So take a look at that article that Wendy will send out. I don't have anything more to add, but I'll change the subject just a tad bit. This is my opinion. This is not JDRF science, but in my discussions with people that have newly diagnosed children or newly diagnosed adults, I encounter lots of people that have type 1 or have been diagnosed. And it is not uncommon to hear someone say, you know, I had the flu, or my child had the flu, and then they had type 1, or they had something happened and they became diabetic. So they're convinced that there's a correlation between the two. And there very well could be. We know that the cause of the immune system could be a virus, something external. But one of the other things that happens, when we're born, we've got more beta cell mass than we need. If you don't have diabetes, you have extra beta cells. And they're there to provide the extra insulin you need for sick days and things that happen. So when you have the flu, for those of us with type 1, you know, when you have a flu or an illness like that, on those days, you're using more insulin than normal. So my theory is that someone has already been in the state of decline in their beta cells, so that they've got autoantibodies, their beta cell mass has been lowered, but they still have enough. They're in stage 2, maybe they're not to that stage 3 yet. So their blood sugars are maybe normal or right on the edge, and then something happens, COVID or the flu, and then it looks like they get type 1. Anyways, that's my theory. That's not the JDRF official line. I'm giving you what I think.
Thank you. And someone also asked in the registration question about if you noticed any trend in adults being diagnosed. The person who asked is 42 when she was diagnosed. And if there's any research for older adults.
Well, I don't know of any research geared towards adults. I mean, you can, when you go to that clinical trials, you can, you know, there are clinical trials that are specific to adults for a wide age range. But I think I mentioned earlier that right now, about half of the people that are diagnosed with type 1 are adults. So it's no longer, if it ever was, a juvenile problem.
Right.
Jude?
And then Mark?
Yeah, for Wendy, I just wanted to make sure you didn't miss it. Yeah. Oh,
Go ahead, Sam. You can.
yeah. So Mark asked in the chat, Jude, can you speculate on projected timeframes for smart insulin encapsulation to go to market? Do you have any information on that for us?
Boy, I'm going to be really careful about throwing any kind of dates out there, because the minute I do, you're going to be mad at me. I really don't. I think there's great promise in the encapsulation project. So it might have been 4 years ago when we were all excited about that first Viaside product. If we could have figured out a way to keep the body from closing it off and killing off the ability to get blood to it, it showed great promise. And then again this year, the first trial of the Vertex line of stem cells. So I'm going to make it real simple here. There's only 2 things we need to do. We've got to come up with beta cells, and we've got to stop the immune system from attacking them. So we've got the beta cell one figured out. So we got half this problem licked. So now it's just a matter of keeping those beta cells alive. I have to think, again, this is my opinion, and I'm going to give you the story on that Vertex. So Doug Melton, who was at Harvard, he's the one that discovered a way to make beta cells from stem cells. He started a company called Sema. He has 2 children. He never intended to become a diabetes researcher, but his 2 children were diagnosed with type 1, Sam and Ella. So he made Sema. He started this company. Well, Vertex, which is a huge pharmaceutical company, bought it last year, a couple of years ago, for almost $1 billion, 950 million, something like that. In my opinion, a company is not going to invest $1 billion into something that doesn't show some great promise. So I'm excited. I think it's not that far off that we're going to see an implantable beta cell device. And then, I know there was a couple questions there about, when are we going to get this a cure for type 1? And I always answer saying, what does a cure look like to you? And it might be different for all of us. For me, if I had an implantable device, every 6 months I went to the doctor and they stuck this little thing under my skin and it would monitor and provide insulin, I'd call that a cure. I'd be happy with that. I know there's other folks who want to take a pill and it cures them of type 1. In my opinion, that one may never happen. It might be years before we can just, take a pill and say we don't have type 1 anymore. But some of these other methods that I talked about here tonight show promise. So I don't know, the encapsulation, maybe within the next couple of years, we could see something that's commercially available.
Thank you so much. Debbie mentioned that she heard about a woman who was diagnosed at age 90. Wow, that's crazy.
Wow.
But she was a recent cancer survivor and had undergone chemo, which had damaged her pancreas. Just thought I would bring that comment up.
So if any of you have ever heard a presentation by Dr. Earl Hirsch, so he's up here at the University of Washington, but he's known worldwide, and he also has type 1. And I've seen a couple different times that he's talked about multiple types of diabetes. So there's more than type 1 and type 2. I know another one's called MODI. And in fact, he gave a presentation on a person who had, they thought type 1, she was insulin dependent for years. And then they figured out it was a different version of diabetes and she was missing some hormone or protein or something, and they figured that out and they cured her type 1. So I've never had the opportunity to ask him, hey, what's that test you took? Because I want to do that, although I don't hold great promise. Anyways, so there's more than type 1 and type 2.
Thank you. Does anyone else have any questions that they would like to ask Jude while we have them these last few minutes? All right. Oh, there was one more about any ties to Apple Watch.
I do know that if you have the do-it-yourself system, the Loop, that it talks to the Apple Watch. I'm not aware that Medtronic or Insulet or the tandem pumps talk to Apple Watches, but I'm not a real Apple guy, so maybe I'm wrong there. But I do know for a fact that the Loop system talks to the Apple Watch.
All right, very good. Well, I think hopefully, Sam, Celia, did we get all of the questions in the chat? We didn't miss any. I will be sharing with you this information and along with a link to the recording. And if you have any questions, Jude has put up on the screen there, as you can see, his email address. So he's happy to answer any questions for you. Please just reach out to him or us, of course, as well, too. Jude, thank you so much for taking the time to put this presentation together. I know you spent a lot of time on it, and we are very grateful. It was so interesting and very promising. And thank you. Oh, looks like there's one more.
It was my pleasure. Thank you.
Quick question. We have some.